Good afternoon, everyone, and thank you for joining us. I'm going to pivot immediately. We have the pleasure of having our Institute Director, Dr. Nora Volkow, with us, and she's going to give some opening remarks.
Happened because obviously the purpose of this meeting is actually to highlight the great science that is ongoing by the Avant-Garde. This is the most prestigious award that the National Institute on Drug Abuse gives to researchers. And its aim is to really push the envelope, to generate science that can advance knowledge in ways that the regular mechanisms will not allow for funding, because it is just sometimes too creative, too transformative. And that's why the Avant-Garde was actually launched, because we wanted to bring, again, the freshness and the new perspectives on our understanding about the intersection between HIV and drugs, and ultimately its consequences to the brain, and how can we use that knowledge for treatment and prevention. So it's a pleasure for me to welcome you all, and at the same time, I do want to congratulate you, because again, this is the most prestigious award that we have at NIDA. So I'm very grateful for your science, and I look forward for your presentations. And Redonna, since we've lost a lot of time waiting in limbo, I'm throwing it back at you.
Okay, great. Well, as I said earlier, we're not going to do lengthy introductions so that we can save time for the presentations and for Q&A at the end. And so I'm going to go ahead and queue up our first presentation by Dolores Albarracín from the University of Pennsylvania, Annenberg School for Communications. And she's going to present on digital mobilization of communities to curb HIV and HCV in Appalachia and the Midwest, theoretical, empirical, and methodological issues. So take it away.
Okay. Can you see the slides okay? Okay.
We can.
Great, great. Well, it's great to see you all, and I'm very happy to be here to introduce this really exciting project to all of you. And I'm going to be specifically talking about digital mobilization of rural communities to curb HIV and HCV in Appalachia and the Midwest. So the story of this is actually probably best told by this video by CDC. So the 220 counties mentioned there were these 220 counties across 15 states that were identified by Michelle Van Hendel and colleagues at CDC. And these are the counties on which my Avant-Garde is actually based. And this, of course, continues to be highly relevant, as you know, because we've only seen this become an even more dramatic problem, with not 47,000 as back then, but 108,000 overdose deaths in 2021, which represented a 15% increase in only 1 year. And this has affected the area in which I'm concerned, as well as the whole nation, more or less, as you can see here, which is showing the increases in overdose deaths. And this, of course, involves the states in Appalachia and the Midwest with which we are concerned. And this has come along with new HIV and HCV outbreaks, some of which are listed here, all associated with drug injection in the region. So this is the really, critical problem that's addressed in this grant, and it was a problem in 2017, 2018, when I designed this, and it continues to be. So the problem is of staggering magnitude, and it also has some really fundamental properties. One is the social causes. These are diseases and deaths of despair, and really related to loneliness, death of hope, the financial crisis that we've seen, the migration of jobs, the job market changes, stigma, not to mention now also this overwhelming pandemic we've been living with for 2.5 years. And this goes along with inadequate communication on harm reduction policies in many of these rural regions that we're talking about. So we have, on the one hand, mixed messages being sent that continue to be sent by mayors of counties, by the media, and that goes hand in hand with inadequate harm reduction policies in these critical states that we're looking at. This was true when this avant-garde began, and it continues, unfortunately, to be true. So if you look at West Virginia right now, and I know we have members who are parts of the communities in this project here today in attendance, there there's been progress. There are syringe service programs that are here appearing in Black, but you see some in white that have been closed down. And it turns out that the majority of West Virginians need to travel between 20 and 76 miles to actually access clean syringes within their state. So this is the critical situation. And the goal of my avant-garde project was to propose a solution which involves creating a network that supports HIV prevention and testing in the counties and support strategies of behavioral change while also reducing isolation, increasing compassion and understanding in the community at large, and doing this through a network intervention that brings together the community at large with people who are injecting substances, because these diseases of isolation are not going to be resolved unless the community really brings them into their care to some extent and friendship. And this is what the project is really trying to address. So what I'm going to do today is talk about the creation of this network, which supports HIV prevention and testing in the counties, and that supports the project. So the project began by creating this grid for the reduction of regional vulnerability associated with drug use and also, other potential community interests. And we built it by first designing a recruitment method so that everything could be done virtually online. We tested this recruitment method in a really innovative and unprecedented way. And we have findings from it, in addition to not just whether it works to actually recruit this network, but also what can this tell us about how communities, stand and work to address these kinds of problems. And then I'm going to tell you about the state in which we are, which is, developing the final intervention that we're going to test and how we'll do that. So this is the network. It involves right now around 85 counties in Appalachia and the Midwest, and counties that were selected by simply selecting 50% of the top 5% vulnerable counties in that study that I showed you living in Appalachia and the Midwest. So these are our target counties. They appear in blue. And what we started by doing was to compile a systematic list of agencies from which we could recruit community agencies and health departments to actually participate in this network. And we actually did it by developing a systematic communication protocol that was implemented by 5 researchers initially within the team. And some members of the team are here. And of course, you can see from the sheer scope that this would not at all be possible without their contribution. So this method has been now tested, validated, and shown to be successful. And it involves a cascading effort that starts with emails to all these agencies, followed by phone calls that lead to then hopefully a Zoom meeting with the agency, let's say health department, prioritizing health departments, drug coalitions, and then all other agencies. So we essentially touched on and communicated with any agency, whether it was concerned with health, but also other areas of interest related to, for instance, law enforcement, drug courts, religious institutions. So we record absolutely every contact we have with them, when it happens, when the outcome is, when the form of communication, who is doing it. And we also obtain recruiter ratings of the contacts, perceived suspicion, interest, knowledge of the situation related to substance use, and concern with it. And then we document the number of contacts and attempts, the number of days that it requires to recruit an agency such as the health department to really be onboarded onto the project, and whether that recruitment effort was successful or not. So we actually did this over a period of several months that then ended with the pandemic arriving, and pre-continuous contacts. And within 5 months, we had 73 counties recruited out of our target of 99. Remember that half that we had randomized to being in the project. So we've actually described then what it takes to be successful in a paper that's published in the Journal of Community Psychology. And we have some knowledge about this. One aspect is there is a certain degree of bias. It takes more context to actually get counties that are within the vulnerable, because they are all vulnerable. The most vulnerable take a little bit longer. And health departments took a little bit longer and more complex to recruit than other agencies. But in the end, success rate was equal across both. And with these data, we're also able to ask some fundamental questions, which we're currently studying by combining these data with experiments and surveys of the counties, about which we have a lot of information. So going on to some findings about what it takes to get authorities to really mobilize and do something on behalf of their counties. One question that we've asked, we were able to actually look at this by looking and pairing up the recruitment data. So did we actually recruit an agency, and how long is this taking, with surveys of the counties, surveys of adult residents within the counties, who answered questions about what their county looks like, what it is to live there. And one of those questions, or several questions, were about community cooperation. So people in my community help each other when needed. People in my community watch out for one another. And we also had other control variables, such as trust in their leaders and community identification. And one lesson that we learned from this is that it takes a certain type of community to make engagement easier. And that community is one that cooperates, and where the residents who survey say, yeah, this community cares, we help each other. In those communities, having that aspect of the extra help in the community, the altruism in the members of that town, actually contributes to ease of engagement. It takes fewer days to actually recruit their agencies, but that also interacts with the vulnerability of that county, implying that when vulnerability is high and cooperation is also high, those are the best conditions to get to engagement by these agencies into the project to advance community health. And you see the same pattern for the ease of engagement measure with the number of communication attempts. So the previous one was days, this one is communication attempts. And again, there is this positive interaction between community cooperation as measured by surveys and the vulnerability to HIV, HCV outbreaks in the community. So we know from this then that we can study how to get agencies to really step up and work towards engaging, the community in better efforts to prevent HIV and HCV in these high-need counties. So at the same time, what Grove does, what this grid does, is it's our community advisory board for the project as a whole, and it also provides our intervention design tasks. So we maintain the board through phone calls and emails to update and discuss member needs. There are 6 RAs currently working as a central contact for between 10 and 20 counties. And by March 2022, we had around 500 interactions with CAB members, over this period that began in 2020, or began in 2019, but 2020 was sort of the full formation. So we also have a monthly newsletter with which we communicate and share what's going on in the regions, sometimes interview CAB members, and we are meeting with the CAB every 3 months with presentations on issues that they care about, and the task force sits within this bigger advisory board. And the task force is now really working at designing what is this effort going to look like, the final intervention that is being developed in the last 2 years of the project. So it's a multi-state intervention design task force that meets bi-weekly and is actually designing part of the content that will go into this network with public health officials and the various institutions within Grove and the community at large, and people who are specifically injecting substances who are all going to be communicating and participating in this intervention. So the intervention will have some more or less regulated, scripted content on sessions related to building community, how to actually get people to empathize and promote what's gonna bring health for everybody. Session on community health, physical and mental addictions, substance use services and treatment, and then getting into the really central topics of overdose prevention and especially HIV, HCV prevention and treatment services, finishing with going back to community goals. And throughout, what cuts across is that altruism and support for science-based policies has to be central to all of these sessions, but they are really behavioral skills, training types of sessions in which people experience what it means to be a community that helps solve problems. So the actual intervention later on that we're working on is being built on Mighty Networks, which is a commercial network platform with interest. It's going to have interest groups and activities that are stemming from those core sessions, moderated games and discussions, as well as email and chat to connect the communities and connect communities within states also. So that's year 1 to 3, and we are now preparing for the efficacy trial, which is a cluster randomized controlled trial with 2 arms, and sampling people who are injecting drugs as well as the community at large, as I said, from different counties within our counties. So participants are going to include adults oversampling for this age range, recruited via phone calls and recruited through our intervention design task force through, various processes, community events, certain service programs, legacy and online media. And we'll have people who are currently using and have injected drugs, and also a cross-section of the community that will be brought together into this really exciting intergroup contact to reduce prejudice and improve policies. And the outcomes, which will be measured every 4 months, will involve testing, remote testing, via, mail kits and verification of PrEP use, syringe exchange, self-reported and whenever possible verified, as well as self-reported outcomes that are broader on condom use, sex exchange for drugs or money, or harm reduction measures, as well as all of the mediators and the data from the platform participation. So with that, that's essentially all we've learned so far, and we remain extremely excited about continuing to contribute to health in these communities. And I'm first of all would like to acknowledge the team as a whole, the network, and also the intervention design task force. So thank you.
All right, thank you so much. We'll open up time for question and discussion. You finished a little early, which is fantastic because it leaves us ample time to have some Q&A and some discussion. So we have 95 participants currently outside of the panelists on the screen, which is fantastic. I've heard we have over 200 people registered, and some people are going to come and go throughout the day. So post your Q&A in the Q&A feature, if people can do that. And then if any of the panelists have questions, they can just come on screen and ask. And so Dr. Varthakavi is going to lead the Q&A, but I'll start while people type things in there. You talked about an intervention that is working with community engagement in sort of a virtual way of doing that and having that occur. And one of the things I was wondering is how you're going to look at addressing health equity within those communities within this virtual platform. And then the second thing has to do with how do you all address stigma against not only people who use drugs, but also some of the interventions, including things like medication for opioid use disorder, but also in some instances, even the provision of PrEP.
Yes, absolutely, Redonna. Those are all central issues. Stigma especially is what cuts across all of the content and every session. We begin slowly with, just generally what it means to be a compassionate community, because that's the opening to stigma. And, fixing stigma is, as you know, not a matter of just saying, well, let's no longer have stigma. Yeah, and so it's creating common goals and contact between different groups and recognizing that we all suffer from similar emotional problems and deal with similar life difficulties, all of us. And that's sort of the starting point to begin to address stigma over each of those sessions. And of course, when we get to services, stigma is everywhere. As you mentioned, it's in the providers, religious ministers. So we're bringing in content and exercises that allow people to at least relate and recognize that and hopefully overcome it. But I think the key feature is the intergroup contact that we generate by bringing the community together with people who are struggling with access to services and seeing firsthand what that means.
Thank you very much for the great talk. And we have some questions that started coming through the Q&As. So there's one from Dr. Don Dolores. Do you have any countries that are shutting down SSPs included in the study? If yes, what is happening with them?
I haven't heard those concerns. It's more, we have more that just never had it, and people have to potentially travel to get these services. So, we cannot address this. Of course, this is just going to be, fixing some or beginning a discussion in a bottom-up way in the community level. We're not able to fix these policies, but we hope that at least in going through this process, people will gain experience in what it means to create something different within a community, and also learning about services and so on, and that will have an impact. But of course, it would be naive to think that this alone can fix everything, but it will at least attempt to address the attitudes within the community. So then if we get the other part, that's ideal.
Yeah, that's an interesting question, given, we also are seeing some shift in policies in some parts of US as well in the recent time. So,
and now I think we really have an opportunity with the White House really being embarked on some real efforts to getting syringe service programs in every county, that in 85% of the counties that are really struggling with overdoses. So this is potentially a really good opportunity for these things to intersect. Thank you, Dawn.
okay. Not seeing any other questions, but so there's another follow-up question. Are you coordinating with NASN or other national groups? I'm not sure what NASN stands for. Don, do you want to, Aaron, can you facilitate, unmute Dr. Gonzalez so he can ask the question?
I see the question.
Yeah.
NASN is the North American Syringe Exchange Network, and it sends out starter kits to people trying to start syringe exchanges. It provides them with information and sometimes small amounts of initial funding, and provides a network for exchanging information, as well as working with the Dave Purchase Memorial Survey annually.
We're not coordinating with them, but I think it would be a real opportunity, because one of the things that will happen through this program is people will can seek and they are making some efforts, like contributing to some of the community efforts. So this could be one of their activities, finding out more about these kinds of programs and grants. Yeah.
Thank you. We don't see any other questions pop up here.
So I'll, as people type in other questions, because I'm heavily involved in the Healing Community Study, which is a community-based study. It is not done in the virtual world, but it is based on the creation of coalitions that develop action plans to be able to address the opioid crisis. I'm interested to know about how you might be, as we have, as the communities have taught us to refer to them as people with living experience, those individuals that are either still involved in substance use or those individuals that are in recovery. And in your case, that includes people that are engaging in behaviors that might place them at high risk with regard to HIV and HCV, or people that are either not engaged in HIV care, but are living with HIV. So how are you all engaging within the coalitions, people with those different types of living experience, to help inform the work of the coalition?
Of the network. So everybody, I mean, as you know, all these agencies and people who are working in these areas are, a lot of them have lived experience, and that's a lot of our task force and a lot of our board. So we didn't have to do anything specific to go seek them out. Simply, seek whatever agencies are working in these areas, and a lot of them bring this experience, and are sitting in our team in different ways.
All right, we don't see any other questions in here. I would like to take this opportunity again to remind the audience that if you have any questions, please go ahead, post them in the Q&A box. That way we'll be able to pass them on to the speakers. Well, if there are none, maybe we should go to the next talk. Thank you so much for this wonderful presentation. And we look forward to great data coming from your study in the next 3 years of the award. We look forward to it. Thank you.
Thank you, and thanks for the support.
Okay, so our next presenter is Dr. Tariq Rana from the University of California, San Diego and the Institution of Genomic Medicine. And his talk is on epigenomic and epitranscriptomic regulation of HIV infection. So thank you very much for joining us, and we look forward to hearing your talk.
Okay, thank you. Let's see if I can share the screen. Okay, can you see it?
I can.
Perfect.
Yeah,
Yes.
that works good. Yes, thank you.
Let me just change the view here so I could see it. All right. Again, thank you very much for the opportunity to share some of our work today. And as I meant here on the first slide, that we are RNA biology lab, and as well as a lot of chemists and biophysicists in the lab. So we study the function and structure of RNA machines in the modulation of HIV and other cellular pathways. And I put the lab address there, so in case you have more questions, which we can answer after the session, feel free to contact me. And this is the most important slide, so I don't want to miss it in case we go over time. So the work was done by these students and postdocs in the lab. Gianluigi, Jason were students, and Chung Chang and Tijan Chow were postdocs. And amazing people to work together with, Cornell University, Chris Mason, UC Riverside, Marcus Call Lab, and Yinsheng Lab, and Chuan He's lab helped us a lot on M6A modification pathways as well. He is one of the leader, and the University of Chicago. And the funding came from, NIDA and all of the institutions. Just brief outline of the talk in this time. Sorry, let me start my timer. So first I'll give the introduction and background of what epitranscriptomic is, what is the RNA modifications. And then part one, I will share the findings, how we went and studied this in HIV and the host pathogen interaction. And second, if we have time, can we learn something more about that RNA modification can modulate in the nucleus, transcription of HIV. So what is epitranscriptomics? So this is the textbook definition. It basically changes in cellular RNA modification, which then end up in changing the phenotypes and affecting causes of this. So although this field is, relatively young, people call it very new, but I just want to give some, historical perspective. So before I start, this is Gianluigi, who had led all these studies on the project of HIV. So eukaryotic RNA has more than 100 modifications. Could you believe it? It's not only one or two, there are more than 100 modifications. And a university at Albany has this, very nice table, if anybody is interested to look at it, what modifications are there. So one may wonder, wait a minute, there are so many, and where to start if one is interested in studying the function of any of these modifications. So let's focus only on these, what we all know. We all know M7G structures, the cap, pseudouridine we heard a lot, 5-methylcytosine and 2'-O-methyl is commonly found and has been studied in host pathogen interactions. And today I will be focusing on M6A. And we'll talk about this in a minute, why M6A. So just to give you the history of this, which I mentioned earlier, I think slide was a little later, that RNA methylation is really not a new phenomenon. People have reported this, you could see in early 70s. And I really salute these biochemists and cell biologists. They did not make a library and send this thing to sequence and got the data. No, they had to do the old-fashioned way, TLCs, P32 labeling, fingerprinting, and they discovered how much HeLa cells and 5', how much mRNA is modified with M6A. And those numbers are true as of today with all these sort of like new methods and new techniques. So let's fast forward 30 plus years now from late 70s, early 70s. So in 2012, these two papers came out. So with the advent of high throughput sequencing and wonderful antibodies coming for any RNA and modifications. So these two groups used a method called MERIP. It's a very simple method. You take an antibody for M6A modified RNA, IP it, and do the high throughput sequencing. And then they figure out this, that there is a 0.5% of A are modified in mRNAs. And the 20,000 M6A sites in mRNA, 95%, and even in non-coding RNA, 7,000 transcripts were modified. And these were the preferred motifs for these sites to add. And M6A is unevenly distributed, on average 3 M6A sites per transcript. And you could see when they analyze the M6A map of density, this is important to see because it's going to come up when we analyze HIV infection. So this is 5' UTR, these are the coding region, and there's a 3' UTR. And these were the motives. So these two papers kind of launched the area in a way that, wait a minute, we could analyze this at the whole genome-wide M6A RNA modification under any conditions. So over the last 10 years or so, many labs have studied, and there is a whole pathway now in terms of M6A. If there is M6A modification in the cell, there must be a lot of other things going on, which will write it, read it, and take away the tag. So here is a simple example. I just, this cartoon in this paper, mRNA, this is a residue. There is a huge complex, more than 1 megadalton. This is, the enzyme is called methyl-3 right here. It assembled with many other protein, it adds methyl group here. As soon as methyl group is added, there are these proteins which read this message. These are YTH family proteins. And you could see that as soon as they bind, they can involve nuclear export, splicing, stability, decay, translation, and many other pathways. And then there are 2 enzymes called FTO and LTBH5. So these are the enzymes which will remove the CH3 and then go back to this one. So this is the whole cycle, and these are the pathways which are involved in this. Now, also, many groups over the last 10 years or so studied what's the role of this. You can see it's involved in so many functions in biology, embryonic development, IPS biology, and you can name it, all these I have listed here. And I think many more will be coming over the years. So we wanted to study RNA methylation in immune function and in host virus interactions. And here is the rationale when Gianluigi started the project. One is that methylation is not something which is added once and it won't be changing, as you see the cycle going through, writers, and there are enzymes which will remove it. And methylation is changing under the different stimuli, environment, and it changes in the stress on the cell. And thus, in a similar way, when any virus infects, they should be changing. And on other strong rationale came from, again, go back 30 plus years. These amazing people have found that our viral RNAs contain internal RNA, M6A modification, including RSV, influenza, and SV40. But the functional relevance of these modifications and how they modulate host pathogen interactions were not known. So we did, this is the first message, HIV infection triggers and reprogram cellular RNA methylation landscape. So just Yeah, I think for these audience, we don't need it, but just a quick overview of HIV life cycle. On a large scale, binding happens. After the binding fusion, RT enzyme takes over. This gets into the nucleus, replication start, assembly, and virus goes on. So we can't study at this stage each step, but we decided to use T cells and then infect with various strains of HIV. So here is the first gel I want you to see. So this is Ethereum stain to show that amount of RNA was loaded equal, control cells and RNA. And this is M6A blot with antibody. You can see there are a lot more changes here. Okay, so I'll come back to the other parts later. So you can see there were changes in M6A levels when there is infection by HIV. Then Gianluigi and other and his colleagues in the lab did the NIDA sequencing, and these are the different motifs and changes. What happens when T cells have been exposed to HIV? First, I want you to see here. So this is the analysis of the whole M6A peaks. You can see that this is control. These are the region going. And with HIV infection, there's a big change in the 5' UTR region and some changes in this area. So meaning clearly there is a big shift in M6A adding M6A being added to the total RNA in the cell. Or at least this contains HIV as well. So these are M6A peak distribution. So this is control, this is HIV specific genes, which I'll show you next. So you could see the changes are happening now. In HIV specific genes, 66% is happening in this region, coding region, went back from 40 to this percent. And only in the regions in introns, there was only 2% in this case compared to 20 and 40%. So these are the peak regions. But then when we look at the motive analysis, very interesting. So instead of this RRACH motive, which is very common for M6A site, there is MGAC motive was found. And these changes were also different compared to the control sample. So there was a M6A being added, and there were the changes in the sequencing. So what are the M6A specific genes in HIV? So we found there were 56 of them. And when you do the analysis for GO, and the top functions were, gene expression of, viruses. And just to give you one example, how we analyze these M6A specific genes. So this is HIV rep sequence. You could see the peak coming from input, control, and how dense this is. That's how we identified 56 genes. And these 56 genes, when you go back to see that many of them are already, previously linked to HIV. These are yes, and these are the, when these genes were published. So you could see that there is a lot happening in the cell, and there are 56 genes that are being specific and being changed by HIV. So now the second question is, okay, so there is change in HIV, change in M6A methylation pattern in HIV genome and in the host, but is this functional? Or meaning does this depend on the enzyme? So in order to test this hypothesis, what Gianluigi did was this. So these are the enzymes, as I mentioned earlier, writer, and these are enzymes which will remove. He picked one of them, methyl-3, because that's the contains the active site, and then ALK-BH5 is one of them, which removes M6A at any site in the RNA. So these are M6A binding protein. So after he depleted these, and you can see this is the control, GP120 levels in T cell. When with two different shRNA, methyl-3 and 14, when he depleted these methylation enzyme and complexes, so he saw that these were coming down in this one. So meaning methylation pathway or methylation enzyme were able to inhibit HIV replication when they were knocked down. On the other hand, when ALK-BH5, which removes this tag from here to here, enhanced the GP120 mRNA. And same then he showed by using the ALK-BH5 B24 Western. So the third message is the HIV RNA is M6A methylated. So you could see now, after he took this gel, he made a probe to hybridize HIV genome and around 9 kb, and you could see here it is HIV band. And by showing mass spec also, he analyzed this, and this is just to show just basically simple, dot plot that HIV RNA was being modified. Then we isolated these RNAs and we sent this to MERIP sequencing, and we found there were 14 peaks which we identified with where M6A was in HIV genome, but most intense were on the 5' UTR and 3' UTR, but we also picked some regions in the middle. And to summarize this, here are what sites in HIV genome and what features could be linked to their function. And then we selected 11 to pursue the function of this and see if this is important. To analyze this, because RRE is a well-conserved structure and Rev protein binds to this to export, viral mRNA. So this is a quick summary of this, so how Rev functions. So early life phase of life cycle, here is Rev protein. Rev is coming out, being translated, goes back to the nucleus and export a lot of RNA, which then goes through translation, replication, packaging, and that's all. So that's the function of this protein Rev. And this binds a well-structured, well-known region of a hairpin shown here is called Rev response element, which is, absolutely needed for its function. So then what Gianluigi did, the experiment was the following, that you made this RRE element, and which should contain these 2 M6A sites at least. And then, okay, so I'll come back to that, why he picked these 2. And then added antibody to this, MERIP sequencing, NRT qPCR, so to analyze if these M6A sites, depend on the activity of methyltransferase complex and the ALK-BH5 activity. So just like the HIV replication cycle, you could see now NTC, there's a knockdown of methylase enzyme, and there is less M6A on these Rev response element RNA. And when there is ALK-B knocked out, there is more on this side coming. Right. So then the next question was methyl-3 ALK-B modulate Rev RRE interaction. Do they bind to Rev protein? So he made stable cell lines of Rev and also, meaning transient transfection by all these different methods. The idea was here now, can we, in this case, for the experiment was that RRE element is modified, but what if this is non-specific and there could be some other RNP complex, which is, changing this activity? So when he analyzed Rev protein specifically, you could see now here, NTC control, methyl-3 knockdown, and then when ALK-B knockdown, you have enhanced binding of this. So what this is showing is there is less M6A and there is more M6A and there is more binding to RRE. Then the final, the next experiment he did was, well, I think, let me go over this. So he picked these 2 regions to modify and analyze their effect on whole nuclear export of HIV genome. So to pick these two regions, there is one other analysis we did. We took 2,500 sequences of HIV from infected cells, from patients, from humans, and then analyze what are the changes rate of these specific sites. Very interesting. So this was modified by, I think, 5% or so, and this one was only 0.25% of the time this was modified. So this is very, very conserved. And then compared to all A's in HIV, this average rate was around 10 to 11%. So he made these mutants specific here. There is no longer A here, and this A is changed, and see which direction this will go. And you could see here now that this is the region where this is the wild type, and this is mutant 1, mutant 2, mutant 3. So mutant 1 is able to export, mutant 2 and 3 is not able to export. And then same thing, will they depend on the activity of methylase enzyme? This is methyl-3, this is ALK-BH5 wild type. Mutant 1 behaved like wild type, mutant 2 and 3 did not behave, and you could see there was very little export there, around 10%. So meaning the conclusion was that this site is the one which is conserved and M6A modification is needed for that. So this is very simplified model of this part is that, there is RRE RNA, which is modified with methyl-3, M6A, then there is more binding with Rab protein, and there is more export, and thus affecting the HIV life cycle. This is very simplified version, and happy to discuss all the other options and possibilities later. So then, this was a kind of highlight of an artist saying that RNA methylation twist of faith in the HIV life cycle. So the summary of this is HIV infection triggers hypermethylation of cellular and viral RNAs. Cellular RNA methylome is reprogrammed upon infection with HIV. And third is methylation of RRE RNA regulates viral nuclear export and viral life cycle. And there are many questions in this area and many more to come. And for example, what's the role of host genes which are modified and how they affect HIV replication? And what are the other peaks? So a few months later, we published these two papers came out in Cell Host and Microbe, and one is eLife. So which also on the functional phenotype, we all agreed on that, and more or less some peaks were conserved in some papers and some we have different peaks in that. But other than the peak, the function of enzyme were same in these papers as well. Then this leads to this idea that RNA methylation during viral infections. Now, there is a subfield, people call it viral epitranscriptomics. So in these three top one, HIV, Zika, and SARS are the ones shown by our lab that RNAs are modified and how they modulate host infection. Now this list really continues to go on: HCV, influenza, HPV, KSHPV, and on. So one postdoc in the lab, Na Lee, actually just published a comprehensive review if you want to see a list of any RNA, any virus, DNA, RNA viruses, how they're modulated, what are the modification sites, which modification. She gave a summary in this article. Now, in the next few minutes, I just want to give you, I have 2-3 minutes left. So update on this part. So one modification, M6A, is very common. And as I mentioned, most of the RNA. And second most abundant modification is called M6AM. So I want you to look at this M6A with this modification. M6AM is, there is a second methyl group at 2' O position of the sugar residues in RNA nucleotide. And we knew this modification is there, but which enzyme does it, that was not known. 2019, 4 groups published these papers, finding. There is a protein PCIF1, they call them CAPM as well. What this is that it showed that there is a sequential co-transcriptional capping of this. Oops. Okay, I'm back. Okay, so I was frozen for a while. Okay, so they showed that there is a co-transcriptional sequential adding of this M6AM formation just at the cap structure. So this is RNA pol2. It starts to make mRNA. So this is going on, and this is C-terminal domain of RNA pol2, which gets hyperphosphorylated at SERV5 position. When this does, there are many other enzymes which adds to the cap and 2' methyl to the cap. They are coming along. And this is where PCIF1 come into action. Just the second nucleotide after cap G called A, this gets modified M6AM. So beautiful work in all these papers. They summarize, how to analyze, how to find these and different methods. So Chongjiang in the lab decided to check this idea in the case of HIV. So I don't have time to go over this. This is published, but I'll just give you the summary. That HIV induces a big decrease in M6AM of cellular mRNA when she compared that. And she analyzed by using CRISPR knockout T cells, more than 2,000 M6AM genes are in T cells and 854 are affected by HIV. And how does HIV do it? If PCIF1 turned out to be that this is the factor which inhibits HIV infection. And VPR, on the other hand, just like the stories of WIF proteins, and they interact with PCIF1 and induces PCIF1 to add poly UBTAC and then go through 26S proteasome pathway. And then interestingly enough, it's a long paper, but there were more than 90 transcription factors which are affected by PCIF1 and HIV infection. And she focused on ETS1 transcription factor, which binds HIV promoter and modulate the life cycle of HIV. So the summary of our findings are here, that there is a ETS1. So this is PCF1, ETS1 mRNA. It gets methylated and it's enhanced the stability of ETS1 mRNA. There is more ETS1, binds to the promoter and inhibit HIV genomic DNA, inhibit HIV transcription. On the other hand, if there is no HIV in the presence of HIV, VPR binds PCF1 and turn into proteasomal pathway, and then there is less PCF1. So there is a list of 90 transcription factors which are effective. Yes. Okay, so I think I'm going to end here. And where is my slide? And take some questions.
Well, thank you, Tariq. That was a wonderful talk. And also, I know you've been not doing well and still made it scheduled to come here present today. So wonderful, wonderful talk. Thank you so much. So we have some questions coming in. So Ashish Sharma asked a couple of basic questions. He says, can RNA be methylated by enzymes involved in DNA methylation like DNMTs? Is there an association between abundance of methylated host RNA associated with HIV latency?
Yeah, I think that's a good question. I mean, the answer is no, because we have analyzed by modulating those enzymes, by knocking out and things like that. So we don't see changes in HIV methylation. Okay, yeah, I mean, that's a good question.
All right, thank you. And hi, Leslie. Leslie asks, in one of your early figures, it looks like regions downstream of the HIV 5' UTR have less total M6A modification. How is this regulated, and why is methylation increased at the 5' end? Does this suggest a role in gRNA packaging, promoter enhancer activity, regulation of latency?
Excellent question, Leslie. Yeah, so I think we really don't know. As I mentioned, that in the field, we all agree that there are 5' and 3' UTR are the major peaks, so we all go after those. But there are some other modifications as well. As I showed in the table, there are other regions which really we need to focus on and study their function as well. Yeah, so I think this could be the case. Thank you.
Interesting. Thank you. We have more coming in. So another question. So is cell stimulation known to enhance RNA methylation?
Again, very nice question. So cell stimulation, T cells, especially, we have done it many ways. And just to give you a quick answer to that is, for us to, also to convince ourselves also that you can stimulate T cells by many other functions, meaning many other ways. We did that. And also different MOI of viruses and different strains as well. So we also confirm that to make sure the sites are conserved in our, functional assay. Yeah. Thanks for the question. Yeah.
All right. So, well, I was going to ask you this. What is the impact of drugs of abuse on RNA modifications?
Absolutely, coming.
Are you going to test that in your system?
Yeah, right. So for that, we have used few drugs, and there is a very big change even compared to HIV. And we also tested plus minus HIV. So there is a very big difference in cellular RNA, right? Meaning because, we are interested in looking at this, how drug of abuse will modulate, host side of biology in the presence and absence of HIV. So now we are pursuing the sequencing and trying to map the sites and which genes are affected. Yeah, thanks. Yeah.
Oh, cool. I mean, I would really be interested in looking at that data.
Yeah.
Because we do have some, loose evidence suggesting that the stimulants do affect these processes very much.
Right. And yeah, I mean, just to add to that, it's not only T cells, we are looking at other cell types as well. For example, we use IPS model for CNS.
Yeah.
And then in that, we also see changes in M6A. Right.
I'm going to take that opportunity to plug in a question that I had because he brought up cell type specificities. Do you notice that, like, with M6A modifications, are there any cell type specificities you would see? Are the types, the genes that you looked at, are they different based on the cells?
Yes. So, I mean, one is that, when you look at overall modification, it's, there are so many transcripts, right? So I think there are two things which are going on. One is each cell may have some common genes which are modified and some which are not common, which are just only for that cell population. For example, we see an iPS-derived CNS cell type, we see different cells. In microglia from iPS-derived, we see different M6A pattern. And same way we see in T cells, for example, like those are different. So I think they're going to be common pathway and there will be different. So it will be fascinating and future fun, studies to look at it, how these modifications modulate those transcripts and their, or in response to HIV or, for example, methamphetamine, like we are looking at, that how they modulate and how they affect those genes and if they have any functions.
Yeah, that'd be good to know because we are really interested in what happens in the CNS space.
Right. And then also, we are, also looking at like a brain from monkey models as well, so just to see that, if they will be conserved. Yeah.
Great. So the next question, which HIV protein is involved in RNA methylation?
Oh, actually, HIV proteins are not involved in RNA methylation. So this is the host complex, which is methyl-3 is actually the key one. And as I mentioned, this is, huge sort of 1 megadalton complex, which contains methyl-3, 14, and a few other proteins. And they are the one which then find the mRNA and then add the tag. Right.
Well, talking about HIV latency, which RNA modifications can be implicated? You may have to take like 30 minutes to answer that, but yes, go ahead.
Yeah, God, that's $1 billion question. So RNA can be implicated. Yeah.
I think the question is that can you talk about, in the context of functional cure?
Right.
Yeah.
So yeah, I think functional cure even to contemplate that, we need to first figure out all these modifications are happening in the context of host modification because we can't separate them out, right? So we really don't know out of those 56 genes which we have identified in host, which are really altered during HIV infection, that how many of them are involved in this phase of HIV life cycle, which is like, being latent versus active or not, right? So we have just really studied only at the active phase. So it's really kind of basically starting the field in a way, you could say, that there were many more studies need to be done and see how latency would modulate these host genes. Everything in HIV modification has to be studied in the context of these 56 genes. But we have studied though, HIV methyl-3 transferase inhibitors do work. They bring down HIV level, they do all the stuff. We could check with CRT and without CRT, and I think those studies are very interesting. But then the problem becomes that how specific they will be for methyl-3 transferase. Because as a question came up earlier, they could also bind for other methyl transferases in the cell. So that's kind of a very interesting pathway.
Absolutely.
Yeah.
So this has to do with the methods in your work. Did you use Jacquard cells or primary T cells?
Oh, actually, we've done both. So we take Jacquard cell and MT4 and few other cell line, and then we go to donors and isolate from T cells from healthy donors. Yeah, we have tested.
In your silencing experiment, did you see any effect on viability or cell functions?
No.
No.
Yeah.
Okay. Great, thank you. So is there any effect of M6 modification of the cap region on TAT function?
Oh,
So
God,
vice
this is
versa, TAD effects on methylation at 5' end.
a
Either
guy
way.
like that. That's my favorite question because, you know, in my like in my past life, I used to work on Tatar. So yes, we like, you know, we looked at that. So, and we also wanted to see that if cyclin T would be affected, and, you know, so the cyclin T and Tatar binding. So far, we haven't seen it. So maybe we need to look harder. Yeah.
All
And-
right. I think there's one more question I'm going to take. So does HIV infection impact methylation patterns in circulating RNAs, example in plasma?
Yes, the answer is yes. Right.
Okay. So you have looked at tissues versus peripheral blood?
Yeah, yes, when, you know, when they say, you know, basically circulation means circulation, you know, from blood and plasma and things like that. Yeah.
Okay.
Right.
Well, there's Pablo Penaloza has a question in regards to RNA modifications, but in the context of mRNA vaccines. Do you know which RNA modifications have been studied in mRNA vaccines besides the pseudo-uridine modification?
Oh God, this is my favorite question, Pablo. Thank you for asking. This is great. Okay, so pseudouridine, I think can be used. So actually, we just published a paper not long ago in SARS-CoV-2. There is a region which is M6A modified. And guess what? When this SARS is modified with M6A, this does not activate all the signaling pathways, for example, you know, RIG-I and all those things. So which means that in mRNA vaccine, it would be a bad idea to put M6A because this will bypass the immune system. It won't activate it. So, yeah, so I think viruses may have evolved these things, and or maybe try at different sites. So I think so based on these few findings here and there, I would suggest that pseudouridine does not play any role to, like, you know, modify mRNA to enhance or dampen down the like an activation response.
Interesting.
Right.
Well, I think we reached the clock there at 2:15. Tariq, thank you so much for fielding all those questions. It was a wonderful talk. And we have scheduled a 10-minute break at this time. I see Jeremy is already on the video, but thank you. I hope you're feeling better. And we will convene after the break and then get going with the next talk that is in order. Thank you all. Great. All right, everyone, thank you so much joining back again after the break. Now we move on to the third presentation on the agenda. This is from Dr. Jeremy Luban from University of Massachusetts Medical School. He will be talking about innate immunity to HIV-1.
Is that working?
Yep.
Can you see?
Yep.
Okay. Thank you everybody for coming. Thank you, Kavi and Aaron, and for putting together this meeting, and certainly to NIDA for funding this incredible program, which has for us has been really an incredible, let's say, inspiration that's enabled us to do things that otherwise might not have been possible for us. Also, I have to say it's an incredible honor to be presenting together with Dolores, Tariq, and Eric, my co-speakers today. So I'm going to give you a couple of vignettes, mainly one story with a little bit of another, and then kind of give you a feel for other projects in which we're looking. I guess the common theme here is innate immunity to HIV, which is kind of in the broadest sense. And I guess Tariq was talking about the host response to the viral RNA. It has all kinds of proteins that interact with the RNA, modify it, respond to it. And I would say that certainly is an aspect of innate immunity to HIV. I want to put some of the experiments I'll tell you about into context, and really in the big context, remind you that AIDS was first reported in 1981, and largely because of foresight and all kinds of research that was done for 50 years prior to that, really miracles happened. So already by 1984, there was evidence for a virus which was cloned that we now call HIV-1. I remember giving, when I was taking care of people who were living with HIV in the clinic, the very first drug we had was AZT. I remember using that as a single agent in '87. And then by the mid-90s, we had our first really effective cocktails, which transformed the pandemic, at least in places where people had access to those drugs. So HIV infection went from being a death sentence to becoming an outpatient disease. So today we have many drugs to choose from that suppress the viremia very effectively, prevent, as a result, the decline in CD4 positive T cells and the immune suppression that's associated with that, in effect, making life possible without progression to AIDS. So one of the issues that we face in this era where we have drugs that are very effective, much of the world now has access, and still we're striving to get the drugs to all of those people who need it. Nonetheless, despite the fact that these drugs prevent the worst complications of HIV infection, they don't cure the infection. And that's because the virus, when it enters the cell and brings its RNA genome into the nucleus, where we now believe reverse transcription occurs in the nucleus for HIV. This is a very recent finding, as opposed to the cytoplasm. The cDNA that the virus makes gets integrated into the host chromatin and becomes a permanent genetic element in that cell. And so that cell, if it's not destroyed by the virus in a active replication, becomes a permanent genetic element. And when people are infected, many infected cells die, but then for reasons that we're still exploring, still trying to understand, some of these CD4 positive T cells survive and go on persisting with an HIV provirus within their genome. And so this is a pyramid showing calculations that have been made by a number of groups looking in the reservoir as deeply as one can. And there still are many questions about exactly what the reservoir is constituted by. Out of the enormous number of lymphocytes in one person's body, some subfraction of those have HIV proviruses. A good 99% of those proviruses are defective. They are incapable of making a productive viral infection. And maybe 1 out of 100 of those can make a replication competent virus. And that reservoir of maybe 1 million CD4 positive memory T cells that persist, that have the potential, each of which have the potential to reactivate active virus replication, active viremia, is a topic of a great deal of effort looking at research today. So for many reasons, including the fact that we now have drugs that keep people alive, there are increasing numbers of people worldwide who are living with HIV, going about their lives. There still are, unfortunately, many new infections occurring each year. And in many ways, the outbreak is changing. And I just came across this one paper, which I thought was very interesting, that was calculating the kind of demographics of the outbreak in the near future. And instead of having primarily younger people who are living with HIV, in the near future, it's going to change a great bit. And the majority of the people will actually be over 50 years of age. One of the things that has been percolating for a while in the clinical HIV world is that there seems to be an increased risk of cardiovascular disease. It's very difficult to measure. It's confounded by all kinds of issues. But along with aging, along with other factors that have been better linked, more obviously linked to cardiovascular disease, it seems that living with HIV infection constitutes its own relative risk for cardiovascular disease. And the relative risk, the magnitude of that is on a par with the better characterized risk factors for cardiovascular disease. Now, there are many possible causes for this, this increase in risk. And a number of the drugs over the years have been shown to alter lipid metabolism. That is a number of the antiretroviral drugs. And I'm just showing you 2 examples of studies that were done looking at associations with obesity in people taking this integrase inhibitor. There were really quite significant changes. And of course, these obesity being a risk factor for cardiovascular disease, this could be a path by which living with HIV leads to increased cardiovascular disease risk. Often with HIV, there are other infections. The main hepatitis viruses, herpes viruses, mycobacterial infections. These creatures cause inflammation of their own right, which could be associated with cardiovascular disease. And then there are all kinds of effects of injection drug use. There are additives that cause inflammation in tissues. They damage vessels where much of the regulation of inflammation occurs. And so these are really obvious possibilities for why cardiovascular disease risk might go up. We've considered some other ones. So I'm going to show you a little bit of data about innate immune detection of HIV transcripts, which we think may be setting off in inflammation, stimulating interferon, that might be contributing in some way. And another theory, which has been, for which there's a great deal of evidence, is that with the primary acute HIV infection, destruction of CD4 positive T cells that are within the lamina propria of the gut. The gut is actually the largest immune organ in the body, if you count up all the cells. With that inflammation, there is often measurable loss of epithelial integrity in the gut, and this is thought to weaken the barrier to our systemic circulation, such that microbial products could leak into the systemic circulation and cause inflammation. And so I'm going to tell you very quickly about some work we've done on innate immune detection of HIV transcripts. And I'm going to tell you about work we're doing on so-called innate lymphoid cells and their role in protecting the gut epithelium and how HIV disrupts them. So we've been working on innate immunity to HIV for a number of years. We have discovered factors, for example, that bind to the capsid of HIV and regulate infection, either in a positive or a negative manner. So cyclophilin A is a protein that binds to the capsid lattice and promotes viral infection. TRIM5 is another factor which associates with the lattice and degrades it. And we've even seen that TRIM5's association with the capsid can activate innate immune signaling, such pathways as NF-kappa B and AP1, MAP kinase, that can contribute to inflammation. We recently have started studying a transcriptional regulator called the Hush complex that in ways that are not clear to us, and we're trying to understand this now, is able to detect the HIV provirus or the proviruses of other retroviruses and regulate the transcription of that complex. What I'm going to tell you about how we believe the unspliced intron retaining RNA that the HIV provirus produces seems to be detected by a receptor that we're zeroing in on. And then I'm going to tell you about some work on innate lymphoid cells. NK cells are actually a subset of innate lymphoid cells, and we've reported some skewing in the populations of NK cells associated with HIV infection that may contribute to inflammation. But what I'm going to tell you about more in this talk are defects in homeostatic innate lymphoid cells that we think are associated with chronic inflammation. So to this story that I've referred to about detection of HIV RNA came from experiments where we were producing dendritic cells from human blood in vitro and challenging these cells with HIV, such that the HIV could enter the cells, reverse transcribe, integrate, and then transcribe. And in response to that process, we found that the cell induced proteins like ISG15, which are critical signposts, indicators that type 1 interferon has been activated. And so we've spent a great deal of effort trying to get at the root of this. What in the virus is detected that leads to interferon induction? And what our work indicates is that the virus, in order to be detected in this way, must complete reverse transcription, must complete integration, must actually generate a provirus in the chromatin of the cell, and it must transcribe from that provirus. So if we block at any of these stages, we don't get this maturation. Of all of the HIV coding sequences, the only one that's required for this process, assuming we establish the provirus, is the HIV Rev protein. And with it, the Rev response element in the RNA that Rev binds to help the unspliced HIV RNA exit from the cell. And the way that works is that Rev serves as a bridge between the unspliced HIV RNA and a host transport protein called CREM1. So if we knock out CREM1 with a drug or genetically, we can prevent this type 1 interferon activation. And taking everything together from wrecking and checking the HIV genome, looking at the host genetics, we've shown, I think, quite conclusively that it's the unspliced HIV1 genomic RNA that is the PAMP, the molecule that is detected as foreign that's activating type 1 interferon. And so a major effort now is to determine the receptor that identifies this And we have a couple of candidates we're looking at. We think this detection of unspliced HIV RNA is going to be of much broader significance, and that it's a way that the host cell has to distinguish self-mRNA, like a standard mRNA shown here schematically, from the more complicated retroviral genomic RNAs, which have these retained splice signals within them. And we think that somehow this structure is recognized by the cell as a danger signal that leads to activation. And one of the things that we're interested in continuing to look at within samples of blood from people who are living with HIV is whether we can see evidence that these unspliced transcripts are indeed contributing to inflammation in human cells and ultimately in vivo. I should point out, this would not require a replication competent virus. It would just require that the structure of the viral genome that's integrated be largely intact. This is just a little aside, but we've been studying another outbreak. There's an epidemic of a retroviral infection in koalas in Australia, which we're learning from. And we see a number of really interesting parallels in terms of the story I just told you with HIV. These poor animals are suffering from this infection, which, unlike HIV, is transmitted vertically as well as horizontally. So it can be transmitted mother to child, and it goes germline. And so individual animals have many, many unique proviruses that distinguish them from each other. So this virus is very actively replicating, integrating into the genome. And we found that an innate defense mechanism that they're generally called piRNAs. This is analogous to a CRISPR system, but in the germ cells of us and koalas, we have this system that in a sequence-specific manner attacks retro elements. And we can see activation of that system in, for example, the testes of koalas. And we can see there, it appears also as if it's this unspliced RNA of the retrovirus corvée that's being detected. So we think this has a much broader significance than simply for HIV. So I mentioned that HIV, there's a lot of evidence that HIV disrupts the epithelial integrity. And Yitao Wang, who was working in my lab, was studying innate lymphoid cells and was wondering whether they may be affected by HIV infection. There's a subset called ILC3s, which make cytokines that maintain the health and the vigor of the epithelium that lines the gut. And he started looking at innate lymphoid cells in the context of HIV infection. So first of all, what are these cells? They are very analogous to T cells. So T cells can be broken down into subsets based on transcription factors and the cytokines they make. Innate lymphoid cells follow very analogous developmental pathways. They're analogous subsets that are defined by the same transcription factors, the same cytokines, but they lack antigen-specific or clonotypic receptors. They're often present in tissues where they serve homeostatic functions. And for example, there's evidence that they protect mice from influenza infection when they're in the lung, or against gut pathogens when they're in the gut. So to identify these cells, it's not a trivial matter. We and others generally gate out all the so-called lineage positive cells. So we add antibodies to markers for T cells, B cells, all sorts of other cell types. And we gate out NK cells if we're looking at just the innate lymphoid cells. There's some positive markers we look at. And in this cohort from San Francisco, looking in the blood, we found that whether people are untreated and are viremic, or people on antiretroviral therapy, or people who spontaneously control, we can see that there is significant reduction in the ILCs in the blood. We see association with markers like soluble CD14 or inverse correlation with markers of CD14, which is a marker of inflammation, inverse correlation with the ILCs. So the lower your ILCs, the higher this inflammatory marker is found to be. And we were fortunate to be able to obtain biopsies from colonoscopies in people who are being screened for cancer, people who are over 50, who are HIV positive. We found we had to look at these samples very quickly after they were obtained, and we could gate on the innate lymphoid cells. And what we found, this is a sample of, I believe, 7 people. These are all people on antiretroviral therapy with undetectable viremia in their blood. When we look in the lamina propria from these colon biopsies, we see very significant reductions in the innate lymphoid cells. And if we look in histology, these are rectal biopsies, we can see that there are indeed, looking at markers that are unique combinations for innate lymphoid cells in these tissues. These green dots, for example, are critically reduced as compared to an HIV negative control. This is a quantitation of the immunohistochemistry from a number of people who donated these tissues. And if we look for markers of inflammation, we see, for example, MX1, which is an interferon-stimulated gene. The levels of this are increased greatly in these biopsy samples. We also see increased markers, this brown stain, this myeloperoxidase, which is a marker of neutrophils, which normally you don't see in an HIV negative gut. This is quantitation of the MX1 and the myeloperoxidase staining in these samples. And clearly, there's much higher levels of these cells and this inflammatory marker in the people who are HIV positive here. So we don't know how HIV is decreasing the levels of these cells. We're quite sure that HIV does not directly infect ILCs because they don't have the right receptors, and we don't detect HIV if we challenge them. So we think the effects are indirect. And we think the effects are probably from certain classes of cytokines that are highly elevated during acute HIV-1 infection. These are JAK3 dependent cytokines. And so we think that probably during the acute HIV infection, there's a cytokine storm that has this ability to disrupt these cells. These changes are present in all the different groups of people living with HIV that we've looked at. It correlates with inflammation, all these different kinds of markers. And we have this sense that these changes are indeed permanent. And there are studies showing that you can perhaps prevent these changes from occurring if antiretroviral therapy is instituted during the acute stage. But once you've gotten through the acute infection with HIV, it may be too late. And I think in the interest of time, I'll just tell you very quickly, we've done similar studies with COVID, and we found really extraordinary associations between innate lymphoid cells in the blood. We haven't been able to look in the lung in this case yet. Our findings are that when you correlate, when you look for adjusting data, adjusting for age and sex, which are known risk factors for severe COVID, you adjust for effects of age and sex on the total lymphoid cells in the blood. The only population that really sticks out as being severely depleted with SARS-CoV-2 infection are these homeostatic innate lymphoid cells. And this is a paper that just went online recently at eLife. So much of the work that I've shown you today was made possible by our funding through the NIDA Avant-Garde Award. We're really very, very grateful for that. This is a picture of many of the people who contributed to this study. And I'll thank you at this point for listening.
Thank you, Jeremy. That was a fantastic talk. We are looking for questions to roll in while that is happening. I was going to ask you if you had a chance to look at any addictive substances in this setting, opioids or stimulants.
Right. In terms of innate lymphoid cells?
Yes.
Yeah. So we've looked into it, let's say computationally. So we've profiled them extensively, and there were a couple of candidates we're interested in, but it appeared as if the levels of the receptors that we could see in our data were quite low in terms of the expression. But it is an important question that we're still very interested in and don't have anything definitive. There are reports of effects of cannabinoids on innate lymphoid cells, though there are some of the expression levels that have been reported, we haven't been able to confirm in the receptors. So we're still banging away at that.
Yeah, I think we would very much like to see what happens in people with chronic substance use.
Yeah.
It would just,
I mean, one of the things that we can do, we can isolate these cells and study them ex vivo. Though studying them in vivo, there are so many confounders, especially in someone who's injecting substances, which can have all kinds of effects. And
right, polysubstance use is a problem.
Yeah.
So we do have like some data, like some published studies showing that opiates actually dampen immune responses, especially interferon dependent. But it would be, good to look at innate immune responses, or what is going on with chronic substance use. We have questions that are just trickling in. Pablo is asking, he's curious about the koala retrovirus. I think you mentioned that it goes to germline. So does it infect sperm and ovules of the animals?
The truth is, we don't know exactly where it's occurring, where the infection occurs. We think it's probably in the very early in the zygote or very early embryo. Based on the proviruses that we've seen, the complexity, it clearly goes germline. Occasionally we see evidence, it looks like there may be some mosaicism, which would suggest that it's that something has occurred past the one cell stage, let's say. We don't think. So there are really interesting differences between males and females in terms of the piRNA system, this defense system that we mentioned. So I mentioned we saw these piRNAs in testes. The fact is that we don't see them in ovaries, and so clearly some of the machinery is not has a
Gender specificity.
Is different, yeah.
Yeah.
So it's very interesting. It's a difficult area to explore. Certainly koalas are difficult to work with. They're a protected species with very good reason. We obtained tissues from animals that had died for unfortunate other reasons. They were not sacrificed for this. It's a developing story, and I think it has all kinds of implications for all kinds of disease going forward.
Certainly.
Thank you for the question. Yeah.
Well, we still got some time, and if anyone has questions, please post them in the Q&A.
Oh, yes, I think I posted on probably someplace wrong. Okay.
Well, you can
Hi,
do
Jeremy.
that.
Well, I
Hi.
mean, again, you know, as always, wonderful stuff. Just a quick question about koala. So piRNAs and PV proteins are involved in line 1 and other sort of like endogenous elements. So were they altered in these studies?
There are endogenous elements in the koalas that are perturbed. There are some cousins of corvée that have been present probably for a very long time, as best we can tell from their sequences. And some of them get activated by the corvée infection transcriptionally, and then it looks like there may be piRNAs against them. There are older elements where there are really well locked in, well established piRNAs that are bidirectional against both strands, against these older elements. There also seems to be a recombinant between corvée and an endogenous element that's also very interesting. So there's a lot of interesting ecology going on in the genome of the koala, clearly.
Right. Great. Thanks.
Okay, so another question here. Why ILC3 are the ILC1 cells not involved in antiviral immunity?
So, a lot of one of the things that we've been somewhat restricted by the tissues we have access to. So in the blood, but far and away, the main population are ILC2s. There are significant numbers we can find of the precursors, ILCPs. There are some ILC1s, but they're very, very few in number. And they seem to have, their markers seem to be a little bit different than what people see in the tissues. We were very fortunate to be able to work with the gastroenterologist who was able to get us fresh biopsy samples so we could look at those. We found, for example, we could not preserve these cells and study them afterwards. We had to look at them fresh in order to characterize them. One can certainly fix them and make sections. That's another story. And that's something that we hope to do going forward, maybe even doing single cell sequencing, but on slides. But in the context of the gut, the ILC3s were the main population that we were detecting in our control samples. So,
would you know by any chance if these cells have receptors for opioids?
Oh, I'm trying to remember. I think we're looking at that in the context of the NK cell subsets, which are a subset of innate lymphoid cell. They're sort of analogous to the way there are CD8 positive T cells with killer function. NK cells are the innate lymphoid equivalent. And there was one subset. It's been a little while since I thought about this, and I don't want to say something out of school, so to speak, but so I believe the answer is yes with one of the subsets at least.
Okay, thank you. No, I believe that's all the questions we have. So thank you, Jeremy. That was an interesting talk. And we thank you again for coming in despite, your positive COVID test this morning. Hope you feel better soon. And we look forward to hosting again your talk another time at NIDA.
Okay, thank you so much.
Thank you. So we move on now to the next, our last presentation for today's venue. This will be from Dr. Eric Verdeen. He's from Buck Institute for Research on Aging. His talk will be on a novel reporter HIV genome to study latency. Hi, Eric.
Hey,
Nice
how are you? Thank
after
you,
a long
Gavi.
time.
It's good to see you, and actually good to see Jerry and Tariq and Dolores. It's been a long time. I guess we've all been cooped up. Also nice to see all of the names on the attendee list. A lot of old friends and a very special community. So I must apologize. I changed the title of my talk a little bit. I'm going to talk about the new reporter virus, but also I'm going to talk about some basic biology of latency. So if I can share my slide, I will try now. Okay, hold on. Let's try again. It's giving me a little bit of difficulty here.
There's a little green box with an arrow upwards.
No, I know where it is, but when the screen comes up, it doesn't show me my slide. Okay, let me see. I think this should be it.
Maybe there are other windows open. Sometimes that helps.
Yes.
There you go.
You see it now?
Yeah, I see the slides. I just have to put them in the presentation mode. And
yes, absolutely. I guess I'm a little rusty here. Usually I don't have any problem with this. I don't know why this all look different. I don't even see the button for sharing. Okay, hold on. For the bar from Zoom is in the middle of it. This should do it.
Yeah, the hourglass.
Okay, do you see it?
Perfect,
okay.
Yes.
Very good. Again, thank you for the opportunity to speak here. I want to start also by acknowledging the transformative power of this Avant-Garde Award, which I had a number of years ago, but really allowed us to really develop novel areas of research. And the presentation today will be consisting in 2 vignettes that actually were directly funded by this program and have had a big impact on my lab and hopefully somewhat on the field as well. So we heard from Jerry and other speakers about the fact that we are now in a great position where we can treat HIV infection, and these treatments have really revolutionized the way we handle HIV. But as has been alluded to also, if the treatment is interrupted, even after a long period of time, in most cases, the virus rebounds very quickly. And obviously this has limited the efficacy of our treatments. If one has to be on drugs for the rest of your life, it's not completely a cure. And so there's been a lot of interest in trying to understand what is the mechanism for the persistence of the virus. And the goals that we are aiming for is either maybe to eliminate the virus completely, or maybe to bring it to a level where even in the absence of therapy, it might still be controlled by the immune system. And there's evidence for both cases. So when we think about HIV persistence, on patients on heart, a number of mechanisms have been highlighted. The presence of a sanctuary has been discussed, particularly in the brain. And so this is the idea that some drugs would actually not access some cells. There's also the idea of a persistent infection in long-lived cells, such as, for example, hematopoietic stem cells, has been discussed as a possibility. And finally, the idea of latency is the one that my lab has been focusing on for the last 20 years. And the idea is that the virus exists in a transcriptional latent state in the memory CD4 T cells, and that following the delivery of an appropriate activation signal, leads to the reactivation of the virus. And we've heard the numbers that Jerry mentioned about 1 million live, fully replication competent viruses being present in most treated individuals. So we've learned a lot, of course, and I'm today here representing a very large field of research and studying HIV latency in CD4 T cells. We know it's established at the transcriptional level and predominantly in central memory T cells. These cells are very rare, which does not simplify looking for them, but still means that the problem is not infinite, and suggests that at some point we might be able to get rid of them. There is evidence that the sequence of these latent proviruses does not evolve, suggesting that there's no low ongoing viral replication, which would really support the model of a true latency versus a low-level replication. And finally, patients that are successfully treated with HAART for even more than 10 years show no appreciable decrease in the size of the latent reservoir, which likely reflects the long life of many of the memory T cells. We know, for example, memory for some vaccination can last, for a lifetime, or for other viral infection as well. So this is probably the biology that underlies this persistence. Now, an approach that has occupied the field for a number of years is the idea of shock and kill, which is the idea that we might be able to eliminate the latent pool of HIV by forcing these latently infected cells to reactivate. So this led to the idea of an LRA, latency reversing agents. A large number of drugs have been shown to have this capability, and we reported this a large number many years ago, longer than I care to acknowledge, that histone deacetylase inhibitors was one of the first, actually was the first compounds that were reported to reactivate latent HIV, and this was in late 90s, and so the idea is that once the virus is reactivated, this would induce, in the presence of a protective treatment against HAART, this would prevent the spreading of the infection, but it would maybe, hopefully, elicit an immune response which would kill the reactivated latent cells. That cell might also be killed simply by cytopathic effect or by CTL response. And so this is what a lot of the field was working on, and so we were really intrigued when we started our funding from NIDA in exploring some of these ideas and in mechanistic details. So today I'm going to show you two different stories that are both related to HIV latency. One deals with the establishment of HIV, and this is the part that will deal with a number of dual fluorescence HIV reporters that we constructed that allow us to study the establishment of latency in primary T cells. And second, I'll be discussing the idea of understanding the molecular mechanism that maintain latency. And this is a large shRNA screen that we conducted a number of years ago that looked for cellular factors that control latency. Let's start with the novel fluorescence virus. So my lab has been working on a number of model systems that allow us to study HIV latency. Some of you might be familiar with the JLAT. This was a JERCAT cells that were latently infected with HIV and that we have used extensively to study mechanism of HIV latency. But we also were always limited by the fact that these studies were conducted in tumor cell lines. Therefore, there was a lot of interest in trying to study this in primary cells. And so to do this, we've generated a number of tools that we call dual fluorescence HIV reporters. And the last product, which was generated during these studies, is called GKO, HIV GKO. And it has a green fluorescent protein, which is replaces the NEF open reading frame, is under the control of the HIV promoter. But right behind it, there's an additional cassette with an outside promoter, EF1 alpha, and another reporter, MKO2. And so cells infected with this virus, immediately after infection, even if there's no viral transcription or viral production, start transducing or expressing MKO2, and so you can therefore detect them. But as soon as the viral production starts being activated, the cells also become GFP. And so using this type of marker, we can define 3 different broad populations that are either productively infected, so these are GFP and MKO2 positive, and we can also detect, even early in infection, a subset of cells that are just only expressing MKO2 and not expressing the virus. And we've done a lot of work, and this has been published in a number of papers, to show that these cells here that we call latent, based on this phenotype, do not express any viral proteins, express very little RNA, and when purified, can actually be reactivated to produce virus. And so this is what we operationally define as a latently infected cells. So since this can be conducted, these experiments can be conducted in primary cells, this has allowed us to really explore the biology of latency establishment. And so one experiment that I want to show you is one in which we did exactly what I'm showing you here, treated them for 24 hours, infected them for 24 hours, then sorted each of these populations, and then looked at RNA quantification in response to a number of latency reactivation agent. And what I'm showing you here is, what I think is one of the most important data that this work led to, is comparing the fold induction of HIV mRNA in patients infected CD4 T cells ex vivo. So these are cells that we isolate from patients that are treated with HAART. We treat them with a number of latency reactivation agents, and we measure the amount of RNA that's produced. And on the y-axis, this is the fold induction that we're observing after infection and purification of the latent cells using the GKO virus. And what you can see is an extremely high level of correlation between what we see in the patients and after infection of primary T cells. And so this has been repeated by a number of other investigators, and essentially establishes the validity of this model to study HIV latency. We've done a lot more work on this, trying really to, actually this is the same data. We've done a lot more work to try using this system to really try to understand what is happening in these cells. Why is HIV establishing latency immediately after infection? And why actually is there, what is the fraction of cells that become reactivated? And in this experiment is an observation that we did not expect. And actually, I'm sorry, this is, I made a mistake. This is the slide before I want to show you. In this slide, we actually looked at the fraction of cells that become reactivated. And we were quite surprised, because even when we used, so again, we purified the latent cells after infection of primary cells with HIV GKO, and then treated them with a variety of reactivation agents, including CD3 and CD28, which is one of the strongest. And no matter which donors, no matter which viral preparation and so on, we never observed more than a few percent of these cells being reactivated. Maximum would be 5 or 6%, as shown here for bryostatin and panabinostat, as an HDAC inhibitor. And so this really raised a really important question, because it suggested that even if we had the most effective agent to induce a shock and kill, the shocking is not very dramatic, and most of the latent cells that we operationally defined as transcriptionally inactive but infected are essentially unresponsive. And this actually, by the way, has been corroborated by experiment that Bob Silicano and his colleagues have done using a limiting dilution assay, and trying to understand what is the fraction of latent cells infected that actually are responsive to these agents. And they're observed numbers of the same order, and suggesting essentially that perhaps shock and kill is not going to be a viable approach. An alternative view of this would be that if these cells do not get reactivated, they might never get reactivated. But I think that was essentially the question, and deciding between the two options is actually quite hard. So we were really puzzled by this, and so we've spent quite a bit of time trying to understand what is the reason for this. One hypothesis was that maybe some of these cells were actually not reactivated. So and in the case of CD3 and CD28, this is actually quite easy because we can look for markers of T cell activation and try to decide whether the population was homogeneous in terms of its activation. And what we saw using markers for CD25 and CD69, which are two sort of globally recognized markers of T cell activation, we found that indeed in the NRLIC, which is the non-reactivated latent cells, there was an increased fraction of cells that were only partially activated. However, the majority of them appear to be fully reactivated. These are the gray cells, CD25 plus and CD69 positive. So even though there might be somewhat of a defect in reactivating a subset of T cells, we still think that there's another issue that prevents these viruses from being reactivated in this experiment. Now, an experiment that we've done separately would be to isolate the fraction of cells that did not reactivate and to try to re-stimulate them a second time. And what's interesting is that time we get another 2 or 3% of the cells being reactivated. And we did the same experiment in Jurkat and as a cell line. So there's intrinsically only at any given time a stochastically determined fraction of a population that will reactivate. And we believe that this is actually in large part due to the site of integration. And to examine this, we have done a lot of work looking at the integration site and host transcriptional activities. For example, looking at, I won't go through all the details of this slide, but there is some degree of correlation between the transcriptional state of the gene in which the virus has integrated and its ability to reactivate. And if you look just on this part here, I believe you see my mouse. The cells, if the gene is silent, we find that the fraction of non-reactivated latent cells is actually much larger. So there's again some degree, some role played by the local environment. We've also spent some time looking at the transcriptional epigenetic marks in these different populations, and again have observed some degree of depletion of activation markers in terms of epigenetic regulation in the non-reactivated latent cells. This is shown here for 2 markers. Now, what is the model that we've established at the end of these studies is the idea that HIV latency is regulated by a variety of factors that can be divided in 2 broad classes. First, T cell activation. So these are activating factors, mostly transcription factors such as NF-kappa B and FAT and many others that are whose abundance is determined in large part by the level of activation of the T cells. In addition, and so obviously if the cells is completely resting, it is much harder to activate the cells. And we know introducing NF-kappa B, an active NF-kappa B in any T cells will lead to reactivation of many of the HIV genome. But in addition, there's another variable which I think is equally important, and it's the integration site. And the evidence that we've obtained is that the integration site can play a very dominant role. Of course, there are some sites of integration where the virus is permanently silenced, and we published this a number of years ago. We found an enrichment of latent HIV integrated in some heterochromatin regions. And so these would be your latently infected cells that can probably never be reactivated. But then there's those that are fully activated, where the transcription site is very permissive to transcription. And then there's the whole continuum in between. And this is why I've argued that latency is essentially sampling the genome, and the outcome in terms of the reactivation or the productive versus latent infection of the virus is a reflection of the integrated product of these two variables, integration sites and T-cell activation. This is a view of latency that is not shared by everybody. Some colleagues have argued that transacting factors are dominant. We feel that integration is a really important component. It also means that there are hundreds of ways in which cells can become latent. And this is likely to really complicate the study and the identification of universal drugs that would reactivate HIV at every single site. Okay, so this would be, and obviously this is reflected in the fact that, 24 years later, after we first reported that HDAC inhibitors reactivate latent HIV, they are still not accepted in the clinic, and their demonstrated efficacy is still lagging. So the summary of this first part is that HIV GKO allows for the quantification of latency reactivation on a cell-by-cell basis. The tool has been used now by a growing number of labs. It's available from Adgene if you're interested in utilizing it. There are more than 20 papers that have used it so far, and I think it's an important tool to continue our studies of HIV latency. We've demonstrated HIV latency is heterogeneous in terms of reactivation. I just mentioned this, and we think the HIV integration site affect the fate of HIV infection. And again, these raise significant question of feasibility of shock and kill. The second vignette that I want to share with you is the focus of our work on trying to understand the factors that lead to the maintenance of HIV latency. And so this is a large shRNA screen that we conducted, in large part supported by the Avant-Garde Award, looking for cellular genes that control HIV latency. And the way we went about this is actually, in this case, using a cell line. And this is one of the JLAT clonal cell lines that Warner Green actually generated in collaboration with us. This is the JLAT that is activated by CD3 and CD28. And on the right panel, so it has a GFP reporter virus. Under basic condition, there's no GFP. You activate it, GFP becomes positive. We can do a dose response curve that you show here. As a function of the amount of CD3 and CD28, we get different fraction of the cells that become GFP positive. And so we used a concentration of CD3 and CD28 that would give us about 50% reactivation, and it will become clear in a second why. Before reactivation, actually, we introduced a very large shRNA library, which was provided to us in collaboration with the laboratory of, I'm blanking on his name, but it will come back. This library was non-validated shRNA. It has about 25 shRNA per gene, targeting about 19,000 genes. And the beauty is that it also has 1,000 different shRNA controls and more than 500,000 different shRNA. And we conducted this screen in duplicate. This reporter that introduced the shRNA also has a mCherry fluorescent protein that allows us to identify the cells that have been successfully transduced. And so what we did, we transduced the library, sorted the cells that were actually contained the shRNA, and then first reactivated them CD3 and CD28, and then sorted the different populations. And you can see we can sort both for GFP and mCherry, and we focused on those cells that were what we call the P2 cells, which are both GFP positive, so they would be reactivated, and they would be also carrying the shRNA. And so what we were able to do then is to isolate genomic DNA from P2 and from a mixture of the group, and do deep sequencing for each shRNA, we can determine whether there's an enrichment in the cells that are activated for any given shRNA. And it's intuitively, you can see that if a shRNA is enriched in these cells, it is likely that this shRNA is participating, or enrich or disenrich. This shRNA participates in the regulation of HIV expression. So we added another layer of complication to this, which is we are, of course, identified a large number of shRNAs that were either enriched or disenriched in the green cells. And we're able to map those in collaboration with Nevin Krogan onto an annotated database of protein complexes, so-called quorum. And so what this allowed us to do is to generate this figure, which shows you a number of protein complexes of which multiple subunits are disenriched. And a good example to look at would be a PTFB on the lower right. So this is a disenriched complex in agreement with the fact that this is one of the major factors that drives HIV expression. And so if it is disenriched in the GFP positive cells, it indicates that PTFB is likely to be a really critically important factor. You can see that we identified other complexes, such as the PRC2 complex on the upper right, which has been previously reported to be involved in HIV transcription. But other complexes also for which there was no data in terms of any role in HIV transcription. And we have focused our initial paper on this one of these complexes called the mTOR complex and TGF-beta, which are interestingly linked. TGF-beta plays a key role in mTOR regulation. And so we have pursued these two together. Just a word about mTOR and why we thought this was particularly interesting. mTOR is one of the key anabolic factors. Its activity is driven kinase. Its activity is driven by nutrients, by growth factors, in particular insulin and IGF-1, and by cellular energy. And it induces, in response to abundance of these factors, it induces cell growth and most anabolic processes, protein synthesis, DNA nucleotide synthesis, and it blocks catabolic processes. So the interesting thing is that there are many small molecule inhibitors for mTOR that we were able to use. And what we were able to show in this experiment using 3 different mTOR inhibitors is that if we treat, so I'll ask you to first look, for example, here on the left, if we treat cells with CD3, latent cells with CD3 and CD28, you can see a really robust induction of latent HIV activation. And we can see that in a dose-dependent manner, the inhibitors all are able to suppress this reactivation. And so this obviously has some new implication, is that instead of maybe using a shock and kill type of approach, we could identify some drugs such as mTOR inhibitors or others. There are a number of other molecules that are being considered. We could actually block the reactivation of latent HIV. Now, the hope by doing this is that we might be able to induce a longer-term locking of the virus in an inactive state. And there are right now a number of research grants and programs that are actually all looking at the possibility of pursuing this block and lock as an alternative to shock and kill. I want to finish by acknowledging our collaborators here. In particular, again, I want to reemphasize the visionary leadership of NIDA in bringing these Avant-Garde Award. I believe that they really have changed the way, the directions of my lab, and I'm really grateful for the support. I want to acknowledge many key members of my lab over the years, over that period, who actually conducted many of these studies: Vincenzo Calvanese, Emilie Benard, Leonard Chavez, Matthew Dahabier, Peter Svensson, and Emilie Battivelli. And I want to acknowledge some of our collaborators, Satish Pillai, who I believe I saw in the audience. Hello, Satish, good to see you. Warner Green and his colleagues, Melanie Ott at Gladstone as well, colleagues at Rockefeller, at UCSF, Stephen Deeks, and the Flow Core at the Gladstone and at Buck. And I will stop here and I look forward to your questions.
Hi, Eric. That was a wonderful presentation. Thank you so much.
Thank you.
I mean, we're still waiting for the questions, but I'm going to take the opportunity to ask you one. So when you looked at the disenriched genes, right? So have you looked at like primary cells or like patient samples? I'm curious if those can serve as biomarkers for latency, any of those. Are there indicators of latency?
Which disenrich? I'm not sure I understood what.
Any of them, any of the genes that you have identified as disenriched genes.
Oh, I see. Those pathways.
Yeah, could you use them as biomarkers to track latency, and have you had a chance to verify that using patient samples?
We haven't done this, and I think one reason that it's a really good question. And we haven't done this directly, concerned by the fact that when you look, and I'll give you an example of why I think it might be very difficult to do this. Bob Silicano, for example, has published a number of papers, and including Tony Fauci, claiming that latency is now driven by integration sites. And so we've been a little bit at a loggerhead in terms of what we find in multiple in vitro models and what they claim to find in patients. And the reason why they do not see an effect of the site of integration is when they're looking at latency, they're not distinguishing reactivatable, non-reactivatable. They're looking at all cells that are, including junk, sort of a terminally integrated defective viruses. And we know, for example, I think Jerry told us the ratio of reactivatable to non-reactivatable is close to 2 in 100. And so when you do studies in patients and you're looking at the global population of all of these viruses that are integrated, you cannot really target those that are truly latent and reactivatable. And I think that's why, I mean, you could look for these markers. I don't know that people have actually looked. I know that there are some studies that are ongoing looking at the effect of rapamycin, another TOR inhibitor, in patients, and trying to see if this changes their course in the long run. Your experiment would be great if we could identify the truly latent cells in the patients.
That is a challenge, isn't it? So the non-reactivated latent cells, cells that never get reactivated. So in your assays, they do contain the replication competent intact provirus, right?
Yeah, I should say that all of our experiments are done with replication non-competent viruses for a simple reason: is so these are ENV minus, but they're complemented in trans by an envelope, which allows us to make viral particle. Once they get into the cell, there's no ENV protein that is being produced. And so they're not infectious. You can rescue an infectious virus by reintroducing ENV, but I have not seen any evidence that suggested that ENV might be playing a role in latency, at least at the level of a non-transcriptional virus. So.
All right, so we have some questions coming in here. So this is like, can you talk about if integration plays a major role then after starting of 5% reactivated cells and making them latent again?
Let me see, I'm not sure I understand this.
Yeah, so only limited cells get reactivated instead of integration is responsible. Okay, so this is from Mudit Tyagi. Can you enable Dr. Tyagi to talk? Maybe he can explain. I think he's asking if in the 5% of cells that you sorted that are not able to reactivate, can you reactivate them.
Here's Dr. Tiagi.
Oh, hi, Eric.
How are you, Mudit?
I'm fine, thank you. Very nice talk. Actually, yeah, sorry about my English is little, not that good. So actually, my point is that, when you sort the cells, you told 5% cell maximum, the reactivated cell. So means those cells has the virus which can be reactivated, means it's integrated at places where it can be reactivated. But if you sort those 5% and put them latent again, and then reactivate, then you find that again, very limited cell get reactivated, not all the cells. So it suggests that integration is not the major player.
I'm not sure, actually. For me, it makes exactly the same point. So so what integration does is that it determines the fraction of reactivation. And we've seen this you're totally right. So we've seen this, for example, with JLAT. You can take JLAT, every single JLAT has a fraction of cells that become reactivated. And you can reactivate them, you can sort the negative and the positive, and you can isolate them. And again, if you reactivate, it will be again the same fraction that will be reactivated. So I'm not sure I follow you, because for me, the data that you're actually pointing to is the evidence that it's the integration site.
Yeah. Okay. So I think that's I think you have answered that. But just I was wondering, let us say if integration is the thing, then in the JLAT cells, you already know where it is integrated, and you know that place can be reactivated.
Yes.
And so if that place allow the virus to reactivate, then after making them latent again, then why is only certain population, or let us say in JLAT only certain population?
Let me try to say this again one more time.
Yeah.
It's not so every integration site imposes on the virus a fractional rate of reactivation. Does that make sense? So some sites are productive. And so, some sites, 2% of the virus gets latent, some sites 10% gets latent, and the reactivation varies as well as a function of the integration site.
Okay, maybe epigenetics playing a role as well. Yes. Yeah. Thank you.
It's not like all or none.
Exactly.
Yeah. All right. Oh, this is something on my mind as well. Like, can you talk something about latency in microglia and how it compares with T cells?
That's a really good question. Microglia in the brain, which is the sort of resident macrophage, have one thing that distinguishes them from your standard macrophage. A latent reservoir to be relevant has to be long-lived, and there is evidence that microglial cells are actually extremely long-lived. And so if the virus were able to integrate into microglial cells and to establish a state of latency, and based on our model, latency is not something that's specific to resting T cells or to activated T cells. Actually, by the way, we find, and this is again another example that demonstrates the predominant role of integration site, is that you can infect activated T cells and you will find latently infected viruses, most likely in very transcriptionally inactive genes. And so if you think about microglial cells, it is likely that if there is significant replication in the brain, and that some seeding might occur that would lead to latency in microglial cells. Obviously, the difficulty of studying this in the brain in vivo is this is not the type of sample that you can get like PBMCs, and I think this has limited in large part. There are multiple models of people who've established latency in macrophages and microglial of different sorts. So I think all of that evidence is there. The hard part is to really study this in situ in the brain of people who are infected, and to demonstrate that it is a relevant reservoir.
Yeah, interesting, because the tissue microenvironment might be quite distinct in brain. All right, so the next question I have here is, how are you differentiating persistence from latency in immune cells in CNS, immune cells and cells in the CNS?
Well, I mean, the difference between persistence, latency is a form of persistence. But latency, at least the way we define it in HIV, is a reversible state of transcriptional inactivation. I mean, persistence is the fact that the virus remains in some form in the infected individual in the presence of the drugs.
So you're basically saying the rules apply, it doesn't matter what cell type it is. It's just the microenvironment be different where they are. That's right. Okay, thank you. And what about cells in CNS? I think there are a lot of questions about CNS.
Yeah, I think, I think I answered it before, and I don't have the, there's a really important area of investigation. If you think about areas of immune privilege, also the CNS, and there are a number of areas where the virus could actually be hiding. Yeah. And right now, in the last few years, people have focused on CD4 T cells because this is a cell that we can actually access, that we can study, and we can, the argument has been, let's try to solve one problem at a time. It could be that there are a number of patients in which it's very hard to detect virus in the brain. And that's not true in every patient. So I can imagine that we're going to see some heterogeneity there as well.
Andrea, do you want to take the mic to ask Eric your last question about persistence?
Sure, I can. Thank you so much. I have a quick question then regarding the last definition of latency as you described it. What I've understood in the literature is that persistence is basically a low level of constant production of virus, while with latency, there's no products being made at all. Is that no longer the actual concept, or am I misunderstanding latency versus persistence?
Latency as to whether this is a complete state of transcriptional inactivation, there is some debate there as well. There are some models of HIV latency where one can detect some early transcripts, which seems to be petering out. There are some, when we looked at latency in clonal cell lines, we saw some clones in which there was absolutely no detectable transcription. And we saw some where there was low-level basal transcription. The key variable, if you think about the virus, is to escape a state of latency, it has to be able to produce TAT, and TAT is really downstream in terms of the viral genome. It's encoded by 2 exons, which is towards the end of the genome. So to produce TAT, you need a full transcribed genome. And there is evidence both in patients and in cell lines that you can have incomplete transcripts that are associated with the latent state. I hope that answers your question.
Yes, it does. Thank you so much.
You're welcome.
All right, I think we have reached the end of questions there. So I want to thank all the speakers for making it for today's symposium. This is, Avant-Garde Award is our flagship initiative at NIDA for HIV program. And it has attracted really terrific investigators in the past and continues to be a crown jewel in our HIV initiative, toolbox. We are really thrilled that this award was able to help you pursue your interest in the HIV space. And we are thankful that for all the work that you have been able to accomplish through this award, and we look forward to many more discoveries that are to come from this funded programs through the Avant-Garde mechanism. And I also want to take this opportunity to thank all the speakers, to also request you that if you have mentees who are early stage investigators who have not yet secured an R01 or R01 equivalent funding, we have a parallel funding mechanism, DP2, it's called Avenir, and it's open for ESIs. And you can check out more details about that on the NIDA webpage. Please encourage your mentees to apply to that. Again, Avenir is open just like Avant-Garde. You know, it attracts research at the intersection of HIV and drug abuse. No preliminary data is required, so please do encourage your mentees to look into that. And I want to thank once again for all the great presentations that we had today. And we look forward to hosting you again at NIDA in near future.
Thank you
And
very
we
much.
keep track of your work. I mean, just letting you know, we keep track of your work all the time, looking for, you know, what's coming out. So we are really proud of this. And thank you. Thank you for making it today.
Yeah. Thank you, Kavi.
And I also
Thank
want
you.
to thank all the audience for dialing in to listen to these important talks and learning a little bit more about the Avant-Garde Award. You choose to be one of our applicants in future, and thank you again for your time.